Okay, 7th grade. Let's continue on with our discussion of DNA, deoxyribonucleic acid. We've talked in the past about how DNA was discovered and how it holds the secret to life. We talked about the history of how the structure of DNA was discovered by Watson and Crick in that story. And we've also talked about the actual structure of DNA. We know that DNA is made up of chains of nucleotides, and a nucleotide contains a sugar, a phosphate, and a base. And there are 4 bases. We've also talked about how DNA is a double helix. It's a twisted helix of nucleotides. So in this lesson, we're going to talk about how DNA works, how DNA helps in making proteins, and proteins are helpful in gene expression. In other words, the traits that a cell has, how those traits are actually expressed from the DNA. Okay, why does a cell need proteins to function properly? Okay, the cell needs proteins because proteins are, remember, the building materials for a cell. Also, enzymes are proteins, and enzymes regulate chemical reactions that occur in a cell. So, how does a cell make proteins? Well, we know there are 2 important nucleic acids involved in this process. There's DNA and RNA, deoxyribonucleic acid and ribonucleic acid. So let's review the difference between DNA and RNA, and also nucleic acids, remember, are responsible for heredity and the control of traits in a body, in a cell. Okay, so DNA again contains the sugar deoxyribose. RNA contains the sugar ribose. DNA is double-stranded, RNA is single-stranded. DNA contains the bases A, T, G, and C. RNA contains the bases A, G, C, and U. There's no thymine in RNA. DNA never leaves the nucleus, but RNA can. Now, there's 3 types of RNA that we're going to be talking about in this protein synthesis. There's messenger RNA, transfer RNA, and we really don't talk much at this level about ribosomal or rRNA. Okay, so the overall process, we're going to right now just go through a real quick overview of the process of making proteins. But basically, there's 2 steps. The first step is called transcription. And that word transcription, you've heard of transcribe maybe, or write down, or copy. So transcription is basically the step in which DNA and its code is copied. Okay. The second step is called translation. So this is where the code is translated into a protein. As you can see, I'm still not very good at writing with this pen and the mouse. Okay, so the code is translated into a protein. So there's 2 steps. And we're going to go through one step at a time on how this actually works. Okay, so we're going to go into this a little more detail. The first step, let's see how the message in DNA gets to the ribosomes. Because remember, DNA stays in the nucleus. It never leaves, and it has the code, the sequence of nucleotides. Now remember, proteins are made out here in the ribosome. So the first step is to get that code or recipe for a particular protein from the DNA here in the nucleus out to the ribosomes so they can translate that code. So here again, here's DNA in the nucleus, and this is DNA's tragedy. It contains volumes of information about making a protein. The ribosomes out here are where proteins are made, and so DNA says here in the nucleus, if I could only get out there, I'd show them a thing or two. So how does the cell solve this problem? It's called transcription again. That's where mRNA comes in. mRNA stands for messenger RNA, and it helps get DNA's message out to the ribosomes. So DNA says, how do I tell those guys what I want them to do? And here's messenger RNA saying, I can help. So the first thing that happens in this process of transcription is that messenger RNA is made. All right, so what happens is messenger RNA is made from the DNA template. So what happens here is DNA unzips. Okay, DNA unzips. The hydrogen bonds between the bases, the nucleotide bases of the double helix, are broken. Okay, they're broken and they open up. And then what happens now is messenger RNA comes in here and it base pairs with complementary bases to one side of the DNA molecule. So here, adenine, remember on DNA, it base pairs with thymine, but remember also that RNA does not have thymine. It substitutes uracil. So uracil RNA comes in and base pairs with this. Adenine base pairs with thymine on the DNA. C to G, U to A, G to C, C to G. All the way through just that section of the long strand of DNA for that gene, for that particular protein. Okay, so it just opens up, DNA just opens up so that it can act as a template for just that section of DNA where that gene for that protein is. So messenger RNA transcribes the code from DNA. So the next thing that happens after messenger RNA transcribes just that section of DNA or that gene code from DNA, DNA zips back up. Messenger RNA then says so long, and it leaves the nucleus, and it's off to go take that code out to the cytoplasm where the ribosomes are. Okay, now let's talk about messenger RNA and this code that he's taking out to the ribosomes. Okay, the DNA's code is copied to messenger RNA in 3-letter groups, or 3 base groups, and these groups are called codons. So the next step, we're done with transcription. Now that messenger RNA is out into the cytoplasm with the code, it meets up with the ribosomes. and messenger RNA travels to where the ribosomes are located. So here they are. Messenger RNA says to the ribosomes, I have a message for you. It's from DNA. So the ribosomes can't really understand the code. So they need somebody to help translate it. The code has to be translated. Now remember, proteins are made up of chains of amino acids. So basically what needs to happen is that code needs to be translated into amino acids so the ribosomes can put them together. So here comes the other type of RNA called transfer RNA. So the ribosomes say, where's the translator? So here he is. Okay, now transfer RNA is like a translator. What happens here is it has two main parts. It has the amino acid up here. This is lysine. That's one of 21 amino acids. And down here at the bottom is a 3-letter code. Okay, is a 3-letter code that is translated into this amino acid. Now this code is called the anticodon. Now remember, the 3-letter code on the messenger RNA is the codon. So the anticodon is the complement to the codons contained within messenger RNA. Okay, so here's messenger RNA right here. Here's the first codon that messenger RNA transcribed from the DNA, UAC. Okay, here is the anticodon to UAC. Remember, this is messenger RNA, this is tRNA. So this anticodon, AUG, is going to base pair with this codon here, UAC. Okay, let's sidetrack here about amino acids again. There are 20 known amino acids. And so how is it possible to get a group of 4 letters to code for 20 things? Well, if you put them into groups of 3, there are actually 64 different codes. Okay, so let's go back to what a codon is. Here's what a codon looks like. It is a sequence of 3 bases. Okay, it's a sequence of 3 bases. Okay, so here's messenger RNA. And GCU is the first 3. Remember, this was copied from DNA or transcribed from DNA. GCU is the first transcribed codon from the messenger RNA, GCU. Here's the second one, ACG. So this is codon 1, codon 2. So this is actually known as the genetic code. And this is how we determine what amino acids go in what sequence. So here's the first part of that gene sequence for that protein. The first amino acid is going to have the codon GCU. Okay, so how do we know what amino acid the code GCU stands for, codes for? Well, there's something called a codon chart, and it's really easy to read. You take your codon, GCU, and you put it in this chart of the 20 amino acids. Okay, so here's leucine, serine, tyrosine, tryptophan, arginine. These are all the known amino acids that there are that make up proteins. Some of these might be familiar to you, like for instance, tryptophan is the amino acid that they say poultry like turkey is high in, that makes you sleepy. Asparagine is an amino acid that is in abundance in asparagus, etc. So GCU, how do we know what that codon stands for as far as amino acids? So you look here at the first letter, the first base, and it's G, and then you come down to the second base here is C, and then the third base here, and you see where they intersect. So let's do G and C intersect here, and then come across here, and U is on this line. So GCU codes for the amino acid alanine, or alanine. Okay, so let's talk a little bit more about amino acids. It's possible for some to have more than one codon, like we saw in that last one with the alanine. There was more than one codon for them. There are 3 stop codons, UAA, UGA, and UAG, and there's also one start codon. Now what these do is they tell the ribosomes to start or stop the sequence. Okay, so this again is showing you how the translator works. First, the anticodons on the tRNA meet up with messenger RNA. Okay. At the start codon, at the ribosomes. Next, another tRNA meets up with its corresponding messenger RNA. Okay, so here we have AUG, the anticodon, meeting up with UAC, and AUG carries the amino acid tyrosine. So if we were to look back at that chart and looked up UAC as the codon, we would find that tyrosine is the amino acid that comes in there. Okay, once 2 amino acids are made, a bond forms between them. This process continues until a stop codon on messenger RNA is reached. So here's the process animated. So there's your protein. Now they are much longer than 3 amino acids long. It goes on, and sometimes some proteins are hundreds of amino acids long. But that's the basic process there. So how does it know when to stop? Okay, then you get to a stop codon, and there are 3 of those, UGA, UAA, and UAG again. So in summary, DNA contains the information needed to make proteins. DNA is too large to leave the nucleus. RNA acts as a set of working instructions for ribosomes to make proteins. And this whole process is known as gene expression. So making the proteins is called protein synthesis, making proteins. It's 2 steps: transcription in the nucleus, translation at the ribosomes, and the whole process expresses the gene.